we were considering the statistical approaches to gene prediction, and we have seen that by using different statistical approaches we can predict the genes. Now we will see some more facts, as we have seen earlier, about the gene code and gene code usage arrays in human. And we also know that the gene codon usage for the coding regions of the human genes, they are different from the codon usage array of the non-coding regions. So by making the difference of these two coding and non-coding regions, we can differentiate the codons present in the coding region. To differentiate between the codons in the coding and non-coding regions, we will take an example of the codons. For example, the codons CGC and codons AGG, they both code for the amino acid arginine. We can see here the codon CGC and the codon AGG. They code for arginine, but we can see that the codon ratio or transcription ratio is very much different. And for the CGC, it is 12 times higher as compared to this AGG codons. So therefore, an open reading frame that prefer CGC over AGG while coding for arginine is likely candidate gene. And we can say that the codon which codes or which have a higher frequency to be coded, they might be considered as the codon that is coding for the sequence present in the coding region. So one can use a likelihood ratio approach to compute the conditional probabilities of the DNA sequence in a window, under the hypothesis that the window contains a coding sequence, and under the hypothesis that the window contains a non-coding sequence. Because we have seen if the codon is coding much higher for any amino acid, then we can say that codon is the codon that codes for the amino acid present in the coding region. So if we slide this window along the genomic DNA sequence, the ratio and the genes are often relieved as peaks in the likelihood ratio plots. Apart from the other statistical approaches, a better codon counter is the in-frame hexamer count. And the in-frame hexamer count accounts for the frequencies of the two adjacent codons present in the coding region. And this approach is discovered by Mark Brodowski, and this is more applicable in the bacterial genomes, because in the bacterial genomes there is no presence of the introns. And the secondly, in the bacterial genomes there are present the specific conserved regions in the sites near the start transcription. But such transcription conserved motifs, they are more elusive in the eukaryotes. So this approach is more applicable in the bacterial genomes, because in the eukaryotes there is a very much complexity of the introns and exons present in the genome. And in the vertebrates, the average size of the exon is approximately 130 nucleotides. And this size of exon is much smaller to be considered, because these small size exons, they do not have a reliable peaks in the likelihood ratio plots while we were examining the open reading frame. And they do not differ enough from the random fluctuation to be detectable. And moreover, codon usage and other statistical parameters, they do not have in common what is going on in actually in the transcription machinery, and that splice the introns.